Hi, my name is Lily Chung, and over the summer, I worked at the Duncan lab at Northwestern University Feinberg School of Medicine, studying senescent cells in ovarian tissue. And this was due to an opportunity from the NIH SenNet CUSP summer internship program. From this internship, I really learned a lot. As an overview, I learned how to adhere cells and maintain cell cultures. And I also learned how to perform immunohistochemistry, or IHC. With cell cultures, I learned a lot about sterile technique and everything I performed with those live cells was performed in a laminar flow hood to ensure that there was no contamination of the cells. I also learned that efficiency and quickness is super important. Every second that the cells were outside of the incubator, we risked losing some cells. And then also consistency was very important. We had to change the media every other day to remove the waste and provide more nutrients for the cells. And consistency can also be performing the same procedures, exactly the same for each flask to remove any external factors affecting the cell growth. For IHC, I took images of cell cultures and I analyzed those images to show viability and see if they're viable to use in further experimentation. To start off, senescence is where cells age and permanently stop dividing, but they don't die off. And a common characteristic of senescent cells is the secretion of senescence-associated secretory phenotype, and that can include things like inflammatory factors. So during my time at Duncan Lab, I participated in 2 projects. This first project that I participated in, we were trying to prove the bystander effect, which is the biological response of a cell resulting from an event in an adjacent or a nearby cell. The overall goal of this project was to use an established model of senescent myoblasts to assess the effects on non-senescent ovarian tissue, and we cultured cortex and medulla from the ovary separately. For this, we use lattice to create a directional co-culture model to maximize the possible effect of a senescent tissue on a healthy tissue. So lattice would filter out the waste and filter in new media automatically. My personal goals while working on this project consisted of, first off, practicing culture techniques with male muscle cells, and then also I optimized conditions for the myoblasts to grow. So trying different types of transwells, different coatings of transwells, different seeding densities, and then at the end, I would practice the proof of concept in lattice. For these transwells, I tested many conditions. First, I grew them in 75 centimeter squared flasks just to get a control. Those were seeded at 400,000 cells per flask. I also tested 24 well plates with no transwells at 3 different densities, 4,000, 6,000, and 10,000 cells per well. There was an old transwell that we tested that's coated with laminin, so I also tested that at those same densities. I tested a new polyester transwell, and we also decided to test agarose molds that would create a 3D model or organoid of those myoblasts. A transwell summary for all these experiments that we conducted. These are all at 10,000 seating densities. This first row was my flasks, and we had been growing those before, and we knew those would adhere, and they did. They did grow a little slower because the passage number of these cells was a little higher, but they still adhered well. The 24 well plate with no transwell also adhered very well. The old transwell that was coated with laminin, as we saw in previous experiments, did not adhere at all. And the new polyester transwell adhered very well, better than any of the other ones that were tested. So that was a positive outlook. And this is the agarose molds cultures. The first row was the agarose molds that we would most of the time use. They're standard molds, and these grew well. And the second row was a custom mold that my mentor had made to help measure the size of the organoids. And at first they grew very well, and they even grew larger than the standard molds. But however, over time, we saw they kind of decreased in size and shrunk a little bit. The second project I work on was immunohistochemistry and histology. The purpose of this was to assess the viability of these cultures and the indication of cellular senescence in the human ovarian explants. So for this, we used 10-day static cultures that, so the ovary was cut into slices, and these cultures were treated with and without 0.1 micrograms per milliliter of doxorubicin for 24 hours, and then they were switched to a doxorubicin-free medium for the next 10 days. And doxorubicin is a chemical compound used to treat cancer and chemotherapy, and we found that at very low doses, it won't kill the cells, but it will initiate senescence in those cells. So I looked at 3 participants for this project. And so for immunohistochemistry, I used CC3 and that's an apoptosis marker. So it'll show whether the cells are alive or dead. And then after we've proven that they are viable, I used P16 and P21, which are cell cycle regulators that can indicate arrest in the cell cycle, and this will show if senescence was induced or not. This is the first participant, RTL 868, and we can see that day 0, the cultures were alive and well, but as time went on with this 10-day static culture, something may have occurred during the culturing, and a lot of the nuclei were not viable anymore. This is the second participant, RTL 872, and this one was actually viable. All of the cultures, day 0 and the ones on day 10 were all still viable. However, the third participant, RTL 886, we also saw problems with the cultures and that they were not viable to further perform experiments on. And this slide is some immunohistochemistry that I performed. This one specifically is for P21. And so it shows day 0. There's no positive signals, as there shouldn't be. And they are very alive and well. The control in the middle row is there's a couple positive signals, but that's just a result of aging and it's still viable. And the ones that were treated with doxorubicin, there are very many positive signals. So this shows that the low dose of doxorubicin was working and was inducing senescence in these cultures. So as a conclusion, and in these, what steps we want to take next for project one, we saw that the cells did not adhere well at all to laminin, and they did adhere very well to polyester, so further experimenting will be done with that in lattice. And then new transwells that are coated with collagen will also be tested to see how well cells will grow on that. And for project two, the 10-day static cultures proved that most of the time, rather than not, they were not viable from further experimenting. So we will section the cultures at day 6, day 8, and day 10 now to kind of show and see what occurred during those 10 days. And this concludes my presentation. I'm so grateful for this experience, and I really learned a lot of invaluable knowledge and information. And this is thanks to the NIH SenNet CUSP summer internship program. I want to thank my mentors, Pooja Devkar, Hannes Campo, and Hannah Anvari. And I want to thank Francesca Duncan for allowing me to work in her lab this summer.